hopefully he gets the email and he starts. Okay, well, let's start right here. Were there any questions about the exam that are burning right now for you, that you just need to know, why did I do X? What did I mean by Y? Something like that, yada, yada, yada. Of course, I left my phone. I'll be right back. I'd like to be on record by saying I detest having to be attached to these things 24/7. I detest it. That's why I don't bring the darn thing with me half the time. Good night. This should not take that long. All right. So today the topic is genetic drift. The genetic drift, we said last time, has to do with sampling errors. Okay, you can load now. Okay, that took way too long. Very good. So we saw last time that you can get these mutations that occur, these particular genotypes that occur in populations, and because of just sampling, one group of them essentially survives, the rest of them don't. And so you get a genetic constitution that is part of an original group. Yes? You want to turn them in now? That way it's off your desk. If you have it, just give it. You want to give it to me later today, that's fine too. And if you have it. If you don't have it, you can give it to me anytime today. I usually go home. No. I go home usually around 6 o'clock, and I'm part of the furniture around here. I'm not hard to find. So, yeah. Just any time. Yeah. Thank you for reminding me. I would have forgotten, definitely. So you have this population. It has a certain genetic variation like that, but because of some whatever environmental something happening or something like that, or just chance or what have you, the only part of it that survives are those that have this particular allele. So even though the original ancestral population of 2 alleles, population that survives by the time you have this many generations, all the rest of them have died, and the only one, the population just has that one allele. So that's a sampling error. And we used that last time by, pulling socks randomly out of your drawer. You might say from that, well, you just have white socks. Well, in fact, you don't have white socks. What you have are lots of colored socks, but because you only sampled a small number, it looks like you only have white socks. So let's say you have a population of doves. Say these doves have white bellies. They're a small population of the doves, but the only doves that survive, say some disease comes through and wipes out all the doves except for the ones with the white bellies, we might think the ancestral species had white bellies. No, the ancestral species had a wide variety. But because of this sampling error, essentially, that population now looks a certain way because you sampled just those particular critters. So this is kind of the picture of a person who's inebriated walking, and alleles can be lost over time or they can be fixed. They have just as much a chance of being lost as they have of being fixed. Genetic drift plays its biggest role in smaller populations than it does in larger populations. That's something that is quite important to realize. It's going to play a much bigger role in smaller populations than it is in bigger populations. So let's use kind of a very mundane example. You have a bag, it's a big bag. You have 1,000 white marbles and 1,000 black marbles. You're going to randomly draw marbles from the bag until 10% of the marbles in the bag, that would be 2,000, 10% of that is 200. Did I do that right? Yeah, 200 marbles. And under normal circumstances, you would expect to get about 100 black marbles and about 100 white marbles. Now, what if you drew 200 black marbles? Is it likely? No. Can it happen? Yeah, it can happen. Chances of this are very small, but it's possible, but it's remotely possible. Now, let's say you have a bag of 20 marbles, 10 black and 10 white marbles, and now you draw 10% of the marbles. That's 2 marbles. What are the chances that you get 2 white marbles? Well, if you were to draw 10% of the marbles from a bag of 2,000 marbles, 1,000 black, 1,000 white, your chances of getting 200 black marbles would be 1/10⁻30th. That's pretty small. What's your chance of getting 2 white marbles if you drew 10% of a bag of 20 marbles, 10 black, 10 white? Now what are your chances? Now your chances are 9/38. That's pretty close to a quarter. Can we agree by looking at those 2 numbers that those are pretty different numbers? So clearly in your smaller population, chance, sampling error, can have a much more pronounced effect than in a larger population. So deviations from the Hardy-Weinberg equilibrium due to chance in small or in moderate populations, that deviation due to chance, that's genetic drift. That's genetic drift. Now, your book has pictures like this. This is essentially computerized This page. Oh, here it is. Page 187 has essentially these experiments. So these are the results of computer simulations where you have 9 individuals and 18 gene copies, and then you just run the simulation and you ask essentially how many of these copies end up getting fixed or lost or what have you. What do you notice essentially from the small population, 9 individuals, as opposed to the bigger one, 50 individuals with a lot more gene copies? What do you notice?
More variation.
Huge variation, a lot more variation. So the oscillations are larger, your alleles are more rapidly either fixed or lost. Several of them show loss, several of them show fixation. And even the others pretty wildly swing from one to the other. When you get to the bigger population, now it's looking to me more like you're really starting to vary slightly more around a mean. So size essentially is going to buffer genetic drift to some extent. This is what you get if you run computerized simulations of populations with various numbers of alleles, using simple Hardy-Weinberg sort of readouts, variation by chance, and then just running it. Is this what you see, in fact, in the wild? The answer is yes. And the experiment, the most famous experiment is also one shown in the book. It's the one that was by Peter Bury. And it's right on the opposite page, page 186, figure 6.4. Please look at that figure when you have time. This is a very famous experiment, Peter Bury's experiment that was done with Drosophila melanogaster. So in this experiment, Bury took 107 experimental populations of the fruit fly, Drosophila melanogaster, and he started with 8 males and 8 females in each of these 107 experimental populations. These flies were all heterozygous for 2 different alleles of the brown allele. So the brown allele changes the eye of the fly from red to kind of a dull brown. So one of them was brown 75, the other was brown 1. Now Bury did extensive experiments to show that neither one of these alleles has any difference in survivability in this type of experiment. So he's growing the flies essentially just in cornmeal, these cornmeal-based media, molasses-based media that I use when I grow fruit flies, which Kelsey was subjected to, and she took genetics from me. And he asked the question essentially, what happened to each of these alleles in these populations? So you're starting out with eight males, 8 females. It's a pretty small population. And from this graph, and this graph shows you essentially, first of all, the number of generations through which you ran the experiment. And then it shows you the number of copies of this particular allele. And so you'll notice essentially that in some of these experiments, this BW75, the brown 75 allele, got lost. In others, the allele got fixed. And so you essentially, after 19 generations, some of them had an even distribution between brown 1 and brown 75. Others, however, had fixation of brown 75 or fixation of brown 1. Those same had loss of the other allele of brown 75 or brown 1, essentially. The first populations to lose Brown 75 appeared in generation 6. And with bigger populations, you wouldn't expect this. This essentially showed dynamics of genetic variation that were not dissimilar to what you're seeing here. Loss and fixation of alleles early, and then loss and fixation of alleles later. So by propagating these for 19 generations, only 30 of the generations, 30 of the generations had lost the Brown 75 allele, 28 had only the Brown 75 allele, they'd lost the Brown 1 allele. Neither allele again had any survival or reproductive advantage. This showed that the role of chance tended to fix certain alleles in the population. Tends to fix certain alleles within the population. Is this the only experiment showing such things? No, not at all. Not at all. So if you, there was another experiment that was done. This was also a laboratory experiment. It was conducted by McComas and Bryant, and it used the domestic housefly. Musca is a domestic housefly. Musca is the one that you chase with a fly swatter. So in this case, these guys, they took wild houseflies, they caught them, and they were able to grow them. Houseflies turn out to be very easy to grow in culture. And he subjected the cultures then to bottlenecks. And so what he did was he took 1, 4, or 16 mating pairs, caught them, cultured them, caught them, and then bottle pairs. So 1, 4, or 16 mating pairs. One, 4, or 16 mating pairs. And they measured using electrophoresis, looking at electrophoretic mobility of particular proteins and the alleles that encode those proteins. They looked at heterozygosity. And they looked at predicted decreases in heterozygosity. Population bottlenecks tend to fix alleles. Because they tend to fix alleles, your heterozygosity is going to go down, down. So which of these populations do you think is going to have the lowest or the highest level of heterozygosity? That one, or that one? Small populations. Small populations, you tend to mix the alleles, so the heterozygosity is going to go down with smaller populations. So that's going to have the lowest, that's going to have the highest. That's what you predict. It's exactly what they saw. Precisely what they saw. Okay, that's in the laboratory. What if you go out into the field? Is this just laboratory observations? Or you start to see things in the field that start to resemble this? Yes, it is. You do see this in the field. So for those of you who like these sorts of critters, this is for you, the greenhouse frog. Greenhouse frog. So this has the rather forbidding scientific name of, spell this right. Eleutherodactylus. This one comes in two different coloration patterns. There's one that is mottled, and there's another that is striped. Now, the eggs of the greenhouse frog, they have to be submerged in water in order to develop properly, but waterlogged soil will work just fine too. So the eggs in water or moist soil. Now, the greenhouse frog has become established in Florida. How did it get established from Florida? It came from Cuba. The greenhouse frogs in Florida, therefore, are a sample of the population in Cuba. Many of these populations are either only mottled or in many cases just striped. You don't see both. Interpret that for me. Josh, go ahead.
I forgot the word for it, but they're like disconnected from each other.
Mm-hmm. So when you sampled a few frogs from Cuba, you had a small population. And one allele got fixed and the other got lost. And that also says you probably had multiple introductions. Since different populations, you have different allelic combinations that give you this different coloration. So that's an example of sampling error. That's just like drawing the marbles out of the bucket and so on and so forth. All right, so this just shows you essentially if T is equal to the number of generations. So at time T equals to N generations. Your frequencies then will be between 0 and 1, but the tendency for them to be between 0 and 1, all these allele frequencies are equally likely. Now, as you start to get fewer and fewer generations, the curve, as you can see here, represents the distribution of these alleles among populations as your population size N gets bigger. Notice what happens as it gets smaller. As it gets smaller, your tendency to fix and lose goes up. As it gets bigger, look what happens. Now your tendency to be between, your not to fix and lose, goes higher. So this again illustrates very nicely and tend to fix or lose, something like that. Okay, this is a really interesting application essentially of this principle. So there's two populations of elephant seals. There are northern elephant seals and there are southern elephant seals. And the northern elephant seals went through a bottleneck. They went through a bottleneck because the baby seals were hunted for their coats. You guys are way too young to remember this, but the baby seals at one time were the way the Inuits would hunt them is they'd find the baby seals and they'd club them to death. And reporters went out and they followed the Inuits around filming this, and they filmed it in the United States, and there was this gargantuan outrage at people clubbing baby seals to death. I must say, when I was a kid and I saw some of those images, they were pretty brutal to watch. The other thing is that there is a Bambi factor, in fact. Baby seals are cute. They're just cute. They're little fur balls. They look essentially like aquatic kittens, kind of, with flippers. So when people see them being clubbed to death, they're going to react. They don't like that. Anyway, this harvesting of these northern seals took the population and went like this, bottlenecked them. So population geneticists said southern population did not go through a bottleneck. The northern population did. We can compare the genetic variation between the southern with that's south. Southern with the, okay, Evan, don't look at me like that. I know which way south. Southern with the northern seal population. That's west, in case you're wondering. I was pointing west when, okay, southern, northern. You know what I mean. Evan's looking at me as if to say, dude, you are in deep trouble. All right, so this bottleneck population, 24 different genetic loci were examined, and all you need to do is you tranquilize the seals and then you take a blood sample. Now, here's a big elephant bull seal. You're gonna run up behind that thing, take a blood sample? And no, I don't think you're gonna get anybody brave enough. So you can tranquilize them and then just take blood samples. And then they wake up and then they're ticked off, of course, but they crawl into the water and you're long gone by then. You don't want to get on the wrong end of these guys. They will, they'll mess you up. 24 different genetic loci were examined. The southern elephant seals, great diversity. Northern elephant seals, no diversity. No heterozygosity found at all 24 loci. That's what bottlenecks do. Small population, you decrease heterozygosity. So all this stuff that we generated in the computer happens in the lab. And when we look in the wild, the same thing happens. So we're dealing with principles that are well established. Fortunately, too, you can look at introduced populations and do the same thing. For example, mynah birds. Mynah birds were introduced to several different areas in Australia, New Zealand, Fiji Islands, Hawaii, and South Africa. Those populations show bottlenecks and reduced genetic diversity. Cattle were introduced to Norway. Again, that's a population bottleneck. If you look at cattle populations in Norway, do they show the diversity of other cattle populations? No. So again, this is really nicely established. Does this happen to humans? Yes, it does. Native Americans, where do Native Americans come from? We'll take a look at this a bit more closely when we look at ancient life, but it's pretty clear that the progenitors of Native Americans came from Asia, crossed over the Bering Strait, through Canada down into North America and South America. So the Amarand populations, they do not have B-type blood. B-type blood is, however, rather widespread amongst Asian populations. So the group that left and went across the Bering Strait down into North America, they're a sample, and that particular sample was small. You had the loss of the B-type allele. So this can happen to humans too. And there's lots of examples we can come up with. I don't want to multiply examples, but I think you get the point. Okay, so this then has very prominent implications for conservation, for animal or plant conservation. Once a species goes through a bottleneck and it starts to lose genetic diversity, you now start to essentially get destabilization of that population. And this essentially shows you through these numbers of these population numbers, essentially, you have essentially the stability, you have exploited, conservation concerns, so on and so forth. And you can start to see here as you have this thing called effective ratio. Effective population size, the census population size in 65 different animal populations. And you can see here, as you go to this side over here, what do you see here? You now start to see exploitation. And now you're starting to see that the number of populations, even though they're going up, is starting to get exploited. And then as your populations, your generations go out, and as this effective population size now starts to change relative to the greater population, now you start getting some holes over here. When your population goes through bottlenecks, you start to lose genetic diversity. Now you start to threaten the ability of that population to deal with environmental stresses. Slight environmental stresses sometimes can now start to take that population and kill it off because it can't adapt. Genetic diversity, as far as we know, is good for populations. Without the genetic diversity, they cannot adapt. All right. So this just shows you essentially using other types of measures, the way your large animal populations, they show greater heterozygosity when you look at migration, looking for diversity using migration in proteins in starch gels. If you look at the nucleotide level, diversity is even greater. We saw that last time. If you have smaller populations, now we start to see 1.1 sort of thing. Same song, second verse, so on and so forth. Now, let's talk a little bit about the implications of this. So in New Zealand, essentially, this shows you the hatching failure as populations essentially go through bottlenecks. So here are your hatching failures, essentially, as the population now starts to decrease. Do you see what I see? Population gets smaller here and gets smaller. What happens to your hatching failure? Starts to go up, doesn't it? Hatching failure here is about the same. Then when you get to, it seems like this right here, that now starts to, you now start to get deleterious mutations that start to become homozygous. So just like what you saw over here with this effective size, that seems to be about the effective size for this particular bird. You now start introducing new individuals. You introduce this number here, and now look at what happens to your hatching failure. You don't introduce enough here, and what do you see here? This is pretty much right in the range of this over here. You introduce fewer than that, so that essentially becomes, that's your effective number. That's your effective number for your population right there. Now, genetic drift, therefore, has led to this theorizing with genetic drift has led to the postulation of this thing called the neutral theory. This is not simple. I have been spending a lot of time this weekend trying to think about how to explain this and make it simpler. And it came up empty every time because it's not simple. It's just not simple. So here essentially is how how it goes. The neutral theory postulates the following. And I'll do this essentially by trying to tell a story and see if the story makes sense to you. If it doesn't, I'll make up another story to see if that makes a little more sense. So you have some sort of population of critters, some sort of critters. Now, with this population, as it grows on the range, as it reproduces, what have you, it is subject to certain selective pressures from nature. It's subject to cold, it's subject to wet, it's subject to lack of food, it's subject to predation, it's subject to all these things. So selection are things that the population has to deal with. However, what's happening to this population molecularly? What's happening to this population molecularly is that you're having random mutations that are occurring within its genome. The neutral theory postulates that the effect on this population of random mutations have more to do with the changes that occur in this population than do selection. And here's where they get this reasoning. Yeah, sure. The random mutations that occur in this population by sheer genetic drift, they play a more significant role in the evolution of the species than does direct selection. Now, the neutral theory does not say that selection does not play a role in evolution. Far from it. The question is which it plays the bigger role, genetic drift or natural selection. You have two schools of thought in evolutionary theory still today. The argument has not been resolved, the selectionists and the neutralists. Have any of you had the great misfortune of reading anything by Richard Dawkins? Any of you even know who Richard Dawkins is? Okay. Richard Dawkins is an evolutionary biologist from England. He's known most famously for his books, which are essentially odes to atheism more than anything else. He is an arch atheist, and he has decided that his job in life is not really to teach evolutionary biology, is to make sure no one believes in God, because as far as he's concerned, there is no God and he hates him. That's Richard Dawkins. If you read books by Richard Dawkins, now, Richard Dawkins wrote, in my estimation, one of the best science books of the 20th century called The Selfish Gene. The Selfish Gene. He has a rare talent for making complicated things simple and understandable. He's a great writer, he's a terrific communicator. Never met the man, but I have some colleagues who have, and they said he is also a first-class jerk. But putting that aside, if you read books by Dawkins, Dawkins is a hardcore selectionist. He talks about how the genome and every base in the genome is constantly being molded by natural selection. So he is as hardcore a selectionist as you will ever get. For example, in his book, Climbing Mount Improbable, he talks about the strangler fig. Any of you know what the strangler fig is? So fig trees, you guys don't know what fig trees are. I had a fig tree out in our vineyard when I was growing up, and we used to pick the figs and eat them, and they're very tasty. There's a type of fig that doesn't just grow as a tree in your backyard and with figs. There's a type of fig, it actually grows like a vine up trees. It surrounds the bark of the tree, and then it grows in to the tree, and it parasitizes the tree. And it digests the tree from the inside out. And over years, there'll be no tree. There's just the vines, essentially, the skeleton of the tree, the branches, essentially, that penetrated that tree. The fig essentially strangles and digests that tree. That's why it's called the strangler fig. Yet this fig is clearly related to domestic fig trees. And Dawkins talks about how the way this tree grows, its very habitat, the way it does things, this was all molded by natural selection. There were forces that essentially drove it to this parasitic lifestyle, and everything about this tree was molded by the forces of nature that, in order for it to adapt, it adapted that lifestyle. And he does that with lots of other examples and what have you. And he is as hardcore selectionist as you'll ever meet. So he's on the selectionist side of the debate. Then there are the neutralists. And the father of the neutralist argument is a Japanese geneticist named Motoo Kimura. And Kimura was about 5'8", and boy, was he feisty. And he would give seminars, and people would stand up and disagree with him, and he'd give it right back to them. He was just, this guy had fire in his blood, and he spoke pretty good English for doing all his work in Japan and living in Japan. He didn't live in this country, he lived in Japan. He spoke pretty darn good English. But Kimura was essentially the father and the greatest proponent of the neutralist debate. So here's essentially Kimura's argument. I'll go through this slowly, try and give you the lay of the land. It's mathematical, but it makes sense once you get it in your head. So just listen first, then write down stuff. I'll go through it again. Listen first, then write stuff down because it's complicated. So let's say a mutation, in order for a mutation to become fixed in a population, it will take a certain number of generations to do so. Let's say it takes 4 times some sort of effective number of individuals. It takes that much for it to become fixed within the population. Takes that many generations for it to get fixed. Now, if this number here, if that effective population SARG is large, then the time required for that new mutation to approach fixation might be huge. It might be so great that another new mutation might have arose during the interim. So let's say the mutation rate is 1 per billion. We get a mutation per billion bases per generation. That's about what it is in humans. So a gene then that has 1,000 bases will have 1 mutation per million bases per gamete, per generation. Okay. So we have so here's our our example here. Put the numbers up here, just so you can get a feel for this. For the frogs. Okay, so we have one mutation. We're going to say there's a mutation that occurs per million. And we have a gene, and our gene has 1,000 base pairs. Thousand base pairs. And the mutation rate's 1 million, 1 per million per gamete per generation. That's just based on mistakes that the machinery makes without any help. Okay, hang with me on this. So if we have an effective population of 1,000 individuals, population size equals 1,000, that means there are how many copies of this gene? It's diploid, so it's 2 times 1,000, so it's 2,000 copies of that gene, right? Make sense? Okay. Then how many mutations should we get over the course of 4,000 generations? Well, we should get 1 per million times 2,000. Because there's 2,000 copies in that population, times 4,000 generations. And that should give us about 8 new mutations over the course of 4,000 generations. Now, there's a constant influx of new mutations. This constant influx of new mutations will increase the variation, but at the same time, that variation will be eroded by genetic drift because some alleles are going to get lost. Now, some mutations will increase in frequency by genetic drift, at least in the long run. And each new mutation has a probability of being fixed, and that probability of being fixed is well established in experiments with genetic drift, 1 over 2 times the effective population. That's the probability of fixation. Probability of fixation. So that means there's going to be a steady substitution of one allele for another. And during this process of substitution, we would expect each genetic locus to be polymorphic. And that assumes that each mutation occurs at a different site in the gene, so that each mutation generates a unique allele and a unique haplotype. And this is sometimes called the infinite alleles model. The infinite alleles model. All right, so that means you have mutations constantly arising. Some will get fixed, some will get lost, but you don't care about those who get lost because you have the introduction of new mutations at a rate comparable to those that get lost. That means you should have polymorphisms at most of the loci at which you have in the in the genome of the organism. Is that what we see when we look at wild populations? Do we see polymorphism as a rule? Yeah, it is. It is. And when Lewontin and Hubby identified a tremendous amount of variation at alleles at different loci of organisms in the wild, they said to themselves, is selection essentially causing this polymorphism? No, that's not how selection works. Selection is usually going to select for one of these. It's not usually going to select for a bunch of them. There's something else going on here. And so Lewontin and Hubby's work that was published originally in 1966, that was thought to confirm Kimura's original thinking. That in fact, while selection played a role, the bigger force molding the evolution of this species was the accumulation of random mutations. And this accumulation of random mutations was taking the genome and essentially creating a genome now that had new alleles being introduced all the time. And those alleles either became fixed or lost, but because they were constantly being introduced, you had polymorphism at most of these different genetic loci. That has more to do with that's a greater force driving the evolution of the species than selection, according to the neutral theory. Now it's time to stop, but let me just pull one thing out of this. If this is the case, that means that changes at loci, you would predict, and we'll get, next time we'll talk about why you'd predict this. Changes at loci will be at rates that are relatively the same. That means you should be able to have a genetic clock that can be used to tell you when organisms diverge from each other based on the amount of genetic change they have. That's a genetic clock theory that falls out directly from the neutral theory. So here's what I want you to do. I want you to chew on the neutral theory and see how much of it you understand. The bit in the book talks about genetic drift. It doesn't get, in that chapter doesn't get quite so much into the neutral theory. The section on bottlenecks is very good and on the founder effect. And then it kind of ends it there. But I want you to chew on the neutral theory and come back and ask me questions. And don't forget to, if you haven't turned these in, turn these in for today, either my mailbox downstairs or my door, or just hand it to me. That works too. And we'll go from there. Okay? Promise? You think about it? Yes? 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 Good. Okay. Come back with questions and then we'll go from there. All right. And we'll see you guys on Wednesday.
Dr. B.
Yeah.
I was just for how can I ask how the reference letter is going?